thanks everybody for being here. It's my pleasure to be here and talk about my work. And thanks for the invitation, Jan and Klaus and everybody at IEEE. So the talk is titled Scientific Integrity Verification Through Image Forensics. I'm Daniel, by the way. And here's the agenda for what I have for today. So we'll go through what is the problem we are addressing, we want to address, and why we care about this problem. Then I'll say how we address this problem and who are we when we say we are working on this, and give some details on our solution, and then what we envision as next steps or future work. Okay, so starting with what is the problem, and we are facing scientific integrity crisis, and we are facing this for a while now. It's not a recent problem. It's been in the academia for a couple of decades now, and to the point that we even have some classic examples of infamous problems. So this is an infamous author. I won't mention the name. There's no need for that. But he used it to be based here in U.S. And in a span of 16 years, this person had dozens of publications that had images like the ones you're seeing here with Western blots. And after these 16 years and millions of dollars of federal funding to their research, people started to notice that the images they had problems, they had manipulations. What pointed that probably the research was not grounded on actual science or actual results. So if you look at, for example, this specific case here, we have these image that you're seeing here. This is called a Western blot, where you have a gel and some proteins are added to this gel and some substances, and then you have an electrochemical reaction. And then the proteins, they react, and you can print this reaction and see the presence or absence of proteins. And by the nature of the process, the substrate, it should be, it has natural noise, so it should not have repetition or patterns across different experiments. But if you look at these particular regions here, you will see that they have the same noise. So it suggests that probably this image here, the blots, the stains that you're seeing here, they were added at a different time than the gel, which is not possible or not acceptable for experiments. And here's an example. So this work was, this paper, and actually this author had at least 8 papers that were retracted. The paper was published in 2002 and was retracted 13 years after the publication. And this is how a retraction, I just want to show you what is a retraction exactly. So whenever you publish the paper in a venue, and after a couple of years, it doesn't matter how much time, if the paper has problems and it needs to be retracted by either the author can ask to retract their work, or the venue may decide, the journal or the conference may decide to retract. What happens is that the paper is kept online, as you're seeing here. So the original paper is still available, but the paper has a retraction notice associated or linked to it. And the retraction notice will contain the title of the paper and have a description of what were the problems of the paper or sets of papers. So this is an example of retraction, which I think is illustrative of how academia proceeds to retract papers. So I'm talking about this now infamous classic example, but we are also seeing some recent cases. So just from last year to 2022, there was this startup that works with investigating drugs or treatment for Alzheimer's, drugs for the Alzheimer's disease. And at last year, they were they were one of the best performing U.S. stocks, getting a lot of funding money. But unfortunately, it seems that some of their reports also had some manipulations, again on Western blots. So it seems they were reusing some Western blots after some cropping. I don't know what was the reason and what was the cause, but some people, they found out that some blots were replicated and they should not. And yeah, we are now in interesting times where we have social media and Twitter and a lot of independent investigators that they really they work, they take their time, their free time, or they busy now their work, and they are spending time finding these things. And there's an entire crowd that helps them to find and denounce the issues, and it's an interesting movement. It's cool to see, and they use the Twitter or other social medias to find problems and make questions and point out things. So yeah, we were facing this crisis. This scientific integrity crisis became more evident or more problematic during the pandemics, right? So in 2020, we saw a great rush of papers papers into publication without proper peer review, and these papers start to be mentioned by everybody from the press to social media, and many of these papers promised some treatments that were not so well based or scientifically based. And as you can see here, writers made their analysis on the problem, and what you see here are all the papers that were published that became were cited in some capacity in the press or in the social media, and all the ones that are seen in yellow, they were not peer reviewed. So we are in these times that we have archive, and papers can get some attention even though they were not reviewed yet. Of course, we have these blog that's Retraction Watch, where they track retractions, and they made some analysis on retraction purposes and made some summaries. And according to them, from the papers that were published with COVID-19, the list of papers that were retracted so far is passing 300 papers. So yeah, with COVID, we had all these issues that are related to scientific integrity crisis, the problem we are addressing. And more recently, we are facing now problems with paper mills. So we have these groups that are producing huge amounts, I mean hundreds of papers, and they may even sell authorship. And the papers they fabricate. So they fabricate the text, they fabricate the images, they reuse the images, and the style is very similar, the way of writing similar. They just change the topics, and we are seeing it's showing up everywhere. And this is a problem we need to mitigate, we need to work with. So this is the problem we have. We have this amount of research that's being fabricated, and it's not actual research, and we need to deal with this. Why we think this is important? The first thing is in terms of impact. And papers that are retracted or that have problems, they have a huge impact on career and reputation. And because we as academics, we collaborate, is a natural thing. Sometimes we don't have full control of our peers. We trust them, right? And it might be sometimes the case that you may not be aware of the entire process of your co-authors, and it needs only one of them to maybe do something wrong. And if you are a co-author, you have some headaches. Just recently, now in June, we had this case that's being discussed a lot, where a researcher, I won't mention the names again, but it seems the researcher collected some information from interviews, and it seems some of the data was tampered from the interviews. And you can see here that this is another blog that looks into these cases, and they made summaries. And you can see here that, for example, they say that one of the primary goals of their efforts to protect the work and careers of younger researchers that have, and we are talking about here 148 co-authors of this particular author, and they are all being impacted by these. And here's what happened, just so if you're wondering, they collected information from interviews, and they noticed that one of the fields where it should have the year, a number, it had a string. And then when they look at these particular cases, they notice that the distribution of the data doesn't follow the distribution of the entire data, in the sense that these particular data with these suspicious answers, they tend to agree with what the author is claiming in their work. And the really sad part here is that these authors in particular, their work is focused on academic dishonesty or academic cheating. So they talk about it. And it was totally unexpected. And I cannot imagine how the co-authors are, dealing with it right now. So that's important to have tools for detecting these problems. Another thing that is more obvious, and whenever we have scientific integrity problems, there's a waste of money and resources, right? Because the research, the science is not good, it's not useful. And also, if you're adding money, you're using money in one place because money is a limited resource, it will be missed somewhere else. So it leads to unfair funding, okay? And finally, the most important problem to me in particular that I think that is the problem with problems that have scientific research that has problems is the ungrounded science. So we have tons of papers that have fabricated data and they claim to have very specific or very special or very good treatments that do not work actually. Medicine or things that, people will spend money and may, use things that doesn't work, and instead of doing a treatment that could work, or you don't even know sometimes what they're doing, what is the effect. So yeah, it's worrisome that some ungrounded science is being produced with these problems. And when you look at, for example, the top 3 most cited retracted papers in Retraction Watch, which is the blog that looks for retractions, you can see here, for example, these 3 papers, and you see the number of citations they had before retraction, and they were highly cited, right? And even after retraction, they're still being cited. What makes you wonder, okay, before I had problems with papers that were already citing this work that was not well grounded, and now I'm still seeing some papers that are still citing those works, even after retraction. So there's a large amount of ungrounded science that is worrisome. Okay, so this is our problem, and this is why we care. So how do we address it? So talking about myself and my colleagues, my collaborators, we are computer scientists that work with computer vision and forensics. And, we know how to use a hammer, so we'll make everything a nail. So this is essentially why we are approaching or addressing this problem by looking at the images and analyzing them, seeing if they have problems. So this might be, the images may give indications that there is some problem in terms of scientific integrity of the publications. And this is useful because images, they are used as proof of results. So you see here, this is a paper from 1952 where folks, they presented the famous photos 51 as a proof of the evidence and of the structure of DNA. So this is an X-ray. And yeah, this is common, especially for biology papers, that they present microscopy or other images as proof of the results of or the outcome of their treatment, the treatments they are proposing, or the outcome of experiments. And it may range from microscopy to Western blots, which is the case I just mentioned, where you can, for papers that work with the presence of proteins, and the Western blots are a way to show that some proteins are present or absent, and they have these characteristics as you're seeing here. They have this gray background and then dark stains, they are fairly easy to manipulate because they have this very well-behaved range of pixel values. And by the way, Western blots, if you take the papers that talk about proteins, in the last 3 decades, these Western blots, they are used in 9% of these papers. So we had a huge amount of papers using Western blots. So yeah, images, they are used in scientific papers, and we can maybe take a look at them and see if they have problems to test the scientific integrity. So yeah, we are in the era of digital imaging, right? And we have editing software, so everybody knows and knows how to use it. It's becoming even easier and easier to use these tools. I'm talking about Photoshop, GIMP, Photo Paint, Inkscape, you name it. And some editings, they are fine, they are not problematic. So here's an example of an edit that's acceptable. So we have here 2 points of views of the same experiment, 2 different spectrums. So first row, second row. And it might be the case that the authors want to merge by using Photoshop or another tool, the 2 layers, just to, improve the explanation of their methods or the outcome of the results. And this is fine, this is acceptable. Other acceptable types are, for example, let's say that you have a microscopy and you want to highlight a specific region, so you may crop that region and zoom it out, so make it larger, and then present your results. So these are the type of additions that are acceptable, not problematic. But in the same way, we have the other types of editions or usages that are problematic and we want to detect. So this is the one that is really common. This is called reuse or repurpose, where you have an image that is the outcome of a previous experiment that is unrelated to another experiment that you reuse the image and present as new results. And it happens a lot. So the author, the first one that I was mentioned, that is now the classic example. It seems he generated some Western blots around 2000, in the 2000s, and in a span of 16 years, he started to reuse all the results and claim as new results. And yeah, this is not acceptable. Other types of manipulations we see is you get a single image and you may remove content or you may duplicate content, and then you can hide objects that don't want to be there. For example, you can remove a cell, or you can duplicate content to claim that your treatment is really working and things like this. So this is not acceptable. You are fabricating your results. And then we see a lot of also what we call splicing. This is when you have groups of images and you take a piece of one and you paste on the other, and you compose your results. So yeah, if you're not clear about it, if you're presenting this as actual outcome of your apparatuses, of your microscopy, this is problematic and you want to detect these things. And if you're wondering if this is a real problem, yes, it is. So here, this is the Office of Research Integrity. This is an American agency that works for the Human Health Services. And they are responsible for doing the investigations, the formal investigations of the authors that got federal funding and had misconduct in their scientific research. And as you can see here, in the time from 2011 to 2015, of all the retracted papers, so this is the number of retracted papers according to investigations they made, more than half of the papers, I'm talking about 67%, they involved problems with images. So many of these works, they had tempered images, they had images that were doctored. Okay, so who are we? Just some quick words, because we are a large group and I'm really proud to be part of it. So we are led by the Purdue University here in the US, and Dr. Delp is the PI. And I'm work at Loyola University Chicago, and I was before in University of Notre Dame, and we have close collaborations with them where we use their computer vision capabilities to help us in solving this problem. We also have some participation from University of Southern California. They're responsible for delivering what we are implementing. We have two Italian universities, the University of Naples, Federico II, and Politecnico di Milano. They are our experts in forensics and signal processing. And also we have a Brazilian university, Unicamp, where we also have their expertise, machine learning forensics. And these are this group. We were initially sponsored by the American Defense Agency and the Air Force Research Lab, and now we are being sponsored by the Office of Research Integrity to do this research. And I'm really proud to be part of this team. Okay, so how do we do it in technical terms? So let's dive into the details. First thing, we developed what we are now calling RIVIEW. It's a system for scientific integrity analysis. As you can see here, that RIVIEW means research integrity is a virtual image examination workspace. So first thing, this is a larger view of the system, and you can see here that RIVIEW has a strong human in the loop nature. So we are not writing a system or developing a system that will replace experts and will be in, let's say, a web crawler and crawl all the papers out there and find problems and, take action. No. This is more of a system that is a tool for the expert, and the idea is that the expert will add information to the system, maybe add the papers, the scientific papers, the PDFs, and the system will give to the expert some evidence to help the expert, the analyst, decide what's going on, and then write a report and maybe present to a jury in a tribunal, or present their report to an investigation. So we are not replacing humans here. We are providing tools to the humans. And here is the five, for the current version of the system, the 5 tasks that we implemented in the system. So it's an end-to-end system that starts with the PDF files and ends with sets of evidence. And I will now just, explain each one of these steps on what we did in each one of them. So the first one is what we call content extraction. And you can see here the system is a web system, right? Where what we can do here, the system allows the user to upload the PDFs of the papers, of the scientific papers. And then the system, as you can see here, automatically extract the figures and make them available in a gallery. And each image is associated to its caption in the paper. So it's really useful for the experts. So they are stakeholders from ORI. And yeah, this is how they operate. So it's really useful for them to do the automatic extraction of the images from the PDFs and know what are the captions, so they know they can quickly read an explanation of the images and know from what PDF the images come. So you can provide as many PDFs as you want and you extract all the images. So this is a very simple implementation. It just goes in the PDF stream and extract the data. So you look into the binary stream of the PDFs and extract the images. Then the next step is what we call panel segmentation. So it is common that some images, they are composed of many panels. So this is the case when you have, for example, figure 1, and then you have panel A, B, C, D. So you have figure 1A, 1B, 1C, 1D. So for the forensic analysis, it's better to get those images that are compositions and segment them into individual pieces. And we developed a solution that is based on computer vision, deep learning, object recognition for doing this. So we segment the image into the modules, the panels, and each panel is classified, so it has a class. So we can classify a panel as either as a chart or a graph or a microscopy or Western blot. So we just, as you can see here, we select images that are composed by many, and we just ask to segment these into the independent panels, and you can see that all the panels are here now independent. So you can now do the analysis on the independent panels. Okay, this is 2 of 5. So the third task is what we call image ranking. This is essentially an image retrieval task. So it's similar to Google reverse image search, where given an image that you have interest, you can select that image and ask the system to rank the other images that you just extracted from the papers from the most to the least similar in visual terms. And it helps the experts to quickly see whenever they think that 2 or 3 images are too similar, you can just, they can get one of those images and ask to generate a rank. And this rank will just have a list of the images from the most to the least similar. And for doing this, we use deep learning, basic description of the image, and we use distance, Euclidean distance, just to rank them. And we do some matching with key points too. So this is a very standard way of doing image retrieval. Then the next step, this is when we start doing forensics. So we have a module where we do what we call copy move detection. Okay, and this piece of the system is developed, and the development is led by the University of Naples, by Dr. Luisa Verdoliva. And the way it works, we get a single image and we generate masks that explain whenever an image has cloned or replicated regions. So just a quick definition, copy move detection is the task of given a single image, it finds within the single image the regions that are replicated. Okay, so we call this operation a copy move or cloning. This is a case here that you see. So this is an actual paper that was retracted. And this paper was retracted because some of these images, they should be all different, but actually some of these panels are the same information. They come from the same microscopy. And if you look at the retraction notice, you will see that in green here, these 2 pieces here, they are the same. So a human found it. Okay, and this is described in the retraction notice. So when we provide this to the system, you can see that the system, the review system, it is able to find that region. So we have a copy move detection piece. The way it is implemented, we use a dense match of image patches. So this is well known in the literature, and we rely on the RGB values and other values of the image, other information to just find repetition. Okay. And Here's another example where there's copy move. This one is with Western blots, and this one is not so evident. But yeah, this paper was retracted too because it has duplications. And in case you're wondering, here's the duplicated region. So these pairs of stains of blots, they were duplicated here. Probably there was no activation in one of them, so it was a composition. You can see the ground truth as based on retraction notice in green, and you can see in red what the system is able to find that replication. So yeah, we can bring this evidence to the human expert and say, hey, we are seeing some repetition here. Should pay attention to that. Here's another example of another retracted paper. And again, some blots here were duplicated. I don't know if you're able to see, but here's the answer. In green, what was pointed out, these lanes are duplicated and they shouldn't. So you're seeing here ground truth in green, and you can see that the system was able to find these. And even the background, it seems these pixels they follow, they have the same values, which is suspicious because again, this should be, they should have noise and should be unique, but that's not the case here. So this image was also manipulated. Okay, this is another example that the system brings. This is what we call, those are false positives. So this is the case where we have different points of view from the same experiment. So we have here, for example, one spectrum, another spectrum, and the authors decide to merge things, right? And of course, the system will find these regions as repeated. And yeah, that's fine. So this is why it's important to have human in the loop in the problem, because the system just provides evidence. It's up to the human to decide and figure out what's going on. And probably by reading the paper, you can figure out that it's not problematic. So yes, the system has false positives. And this is why we advocate that it shouldn't be fully automatic. We need an expert using this. Okay, then we have the copy move detection, and then we have the last piece of the system, which is provenance analysis. And just to help you, provenance analysis is a problem that we address in forensics, in image forensics, where we wonder if given an image, we try to answer if that image is real or it was manipulated. So here you have a classic example now, it's been out there for 5 years, where you have these supposed meeting of UN, and it's portraying, putting in the center, and other leaders paying attention to him as if he was powerful or important. And of course, when you use provenance analysis, you can find the pieces, the other images, they are the source of this one, that is a composition. And provenance analysis will help us to find these images, and let's say in the web or in a data set, and also build this graph that you're seeing here that explains that, okay, this image is not real, it's a composition, it has a host, which is the case here with empty seat and United Kingdom instead of Russian Federation. And here's the donor, so Putin was in another picture and he was spliced here, he was pasted, and this is a composition. So this is the problem of provenance analysis. If you want to know more about it, we have a book chapter in this multimedia forensics book that is open access, where we present all the different techniques for doing image provenance analysis. What we did here in this research, we are applying provenance analysis to the figures that come from the papers. So this is an actual case again. So this is a retraction notice that involves 7 different papers. And you can see here that one figure from, I suppose, 2002 was used across other 6 different papers, claiming different results. So not mentioning at all that it was an original experiment done a long time ago. So these results were presented, these Western blots were presented as new results. So you can see here. And this is what is described in the retraction notice. So for this particular case, when we presented to the system, we uploaded the 7 PDF papers, extract the image, and ran our provenance analysis piece. This is the graph we obtained with the system, okay? And you can see here the graph we got was more complex than the one that we have in the retraction notice. But you can see here that in green, so these pieces, they are the same as here. So we were able to detect these true positives, right? So we were able to find these regions, and they were documented in the retraction notice. So for example, if you take a look at the pair AB here, this is what the system found. So you can see that the authors in paper 1, they reused these graphs and the Western blots in a different style, in a different layout, but they just replicated the content as if it were different experiments. And you can see the system was able to find, and we could find these edge in the graph. But then comes the question, okay, what happens for the cases where we have in red? Do we have false positives, or were they missed or not seen by humans? And yeah, so we took a look at another pair, one of the red ones that was not documented in the rejection notice. And you can see here that when we take node N and node D from paper 3 and 4, you can see that, yeah, they share information, and the interesting and sad things that in paper 4, the information is the same after a flip. So it seems the authors, they flipped the image. As you can see here, it's flipped, and they reused the image claiming new result. And the system was able to find it because, of course, the system, it's a computer, so it's easy to compare things. But for a human, yeah, I don't think they were not able to see it, so that's why it's missing the rejection notice. So this false positive is actually not a false positive, but something that maybe a human and the experts missed. And it's great that we have now the system, because now we can find these situations and bring to the humans' attention. Okay, so we developed this system, and then we had the situation, okay, we want to write a paper about it. We want to bring or make the system available to the community, and we want to have collaborations, and also we want other people to work on this problem. So how can we how can we evaluate the system quantitatively? And the solution for us was, okay, let's establish a benchmark. Let's get all these rejected papers, let's annotate them, and let's make them available for the community. Not the papers, but at least the pointers, because we don't have the copyrights of the papers, of course. But we are opening the annotations. So this is what we accepted as a challenge. We built a data set of papers with images that had problems. So for these, we generated a group 1 that had 298 retracted papers due to the problems with images, and these rejection notices are well documented, and we could use them to generate annotations. And then following how ORI operates, the agency that funds us, we took the authors that were involved in these retracted papers, and we got more 358 papers from these authors, because there's a high chance these papers also have problems. So with these, we created a group 2 and added to the data set. And then because we are interested in knowing how the system deals with false positives, if it's too high, or we are pointing out a lot of evidence that is not worth. So I just decided to go out there and get some papers that are supposedly clean, because we cannot guarantee, right? But so far they were not involved in any investigation, and we collected more 332 papers. And for this set of almost 1,000 papers, we provided task-wise annotations. So for each one of the 5 tasks that I just mentioned that we did in the system, we provided annotations for each one of them, okay? And here's some details of the annotation. So for the first task, for example, that is the one for extracting the figures from the PDFs and linking the figures to the captions. Yeah, we took 1,800 figures and associated them to their respective captions, and this involves 285 papers, and we made an annotation for these, expecting that other researchers will try to do the same task and compare to our results. For the task of panel segmentation, we also took some images, 3 figures, and we just annotated segmentations, the expected segmentation, so we generated masks of how the panel should be segmented, and also we classified these panels. And for the task of image ranking, which is the one where it's similar to Google reverse image search, where we give an image and get a rank. So we selected 2,000 and almost 3,000 panels, and we generate for each one of them ranks from the most to the least similar images, and we expect systems to give their answers and compare to our ground truth. We also provided annotations for copy move, so we read the rejection notices from 125 papers, and for 180 figures, we built masks that they activate where the regions are replicated. So the masks, they show for a single image what are the regions that are duplicated, that are cloned. And finally, for provenance, we need the graphs. So we took 85 papers, read the rejection notices that explain reuse, and then we built 70 graphs. And the 70 graphs, they involve 591 figures. So these graphs, they explain how one paper uses the image of the other paper that may use of the other one, and so on. So we have these graphs explaining usage. And this is what we are making available. So a data set of these annotations. And also we propose some metrics. So for example, for image retrieval, we took the system as it is implemented right now. We decided to report results in terms of image recall. So how good are we in getting the images that are expected to be extracted from the papers? And also how good are we in getting the captions? So we compared the captions with Leverstein distance to see if the same caption, and also BERT score to see if the semantic follows the same. And you can see here, yeah, the perfect here should be 1, and you're seeing here the numbers we are getting were not perfect, but it seems it's still useful because our eyes is excited with these results. So yeah, this is what we get for the first task. For the second one, panel segmentation, we advise or we suggest to use the intersection over union to given the ground truth mask of the segmentation and the mask that is returned by the system, we compare these masks and see if they match. And we expect a perfect match as 1 in this case. And you can see here that we are far from 1, but it's still useful results. For image ranking, we observed the precision at top 1, at top 5, at top 10 images. And you can see here that we are far from being perfect, but still we get related images. So this is some work that we need here to make improvements. And for copy move detection, following the literature from forensics, we use the F1 score, which is the harmonic mean between precision and recall of the classification of the pixels of the masks that we generate when we classify them as either pristine or duplicated. So you can see here, we are far from 1, but still it's useful. It produces useful results. And finally, for provenance, following DARPA's and NIST's metrics, we use what we call vertex overlap, edge overlaps, and vertex and edge overlap. And we use these metrics just to compute how the graphs that we have in the ground truth and the graphs we generate, how they overlap. So you measure this in terms of the vertices only, or the edges only, or both, a combination of them. And you can see here that we get, for example, 64. It's not exactly percent, but it is it's similar to F1 score of getting all the vertices of the graphs. And you can see for edges, we're not that great, but in the end, yeah, we do well for finding evidence. This is the important thing here. We want to find evidence that maybe humans had missed things. And this, all that I just talked about is in a publication we published last year for scientific reports. The system has had another name. It was called SILA before, but we had to change because of copyright issues. And yeah, if you want to go there, the paper is available, the modules in the coupled modules are available on GitHub, and you can download them, get the names of in the data set, right? So you can get the pointers to the DOIs of the papers, download them yourself, and then do your experiments. And then what's up next? So, okay, this is what we are doing with this research so far. So what remains for future work and collaboration? So first thing, if you look at the results, we have all this space to improve. So all of these that you're seeing in red is opportunities for getting better results, and look forward to see more folks join our, our endeavor and use the benchmark and improve these numbers. And also, we have an implementation of the system. Right now, we cannot make this available, and I'm happy to answer any questions you may have about it, but we make the coupled modules available. And the system, it has a client-server architecture, as you've seen here. And yeah, there's so much space for improving the graphical user interface and how results are shown. And as you can see here, we are proposing an architecture where each module is a Docker container, so it's independent, and we have a queuing server just to queue the requests and attend requests. But yeah, there's a lot of space here for implementing other ways of using these containers and doing better interface and better system. Also, I have some criticism on how we are retracting papers right now. The way it works, the investigations is really good, and it takes quite a time, and I understand why it takes a long time. But once a paper is retracted, let's say you have all these papers that are citing each other. Let's say that this paper was retracted. It just stays like this. So you create a retraction notice, and there's no work after that to track what are the other papers that maybe should be flagged or should be mitigated or should openly treat or make corrections because they are citing retracted work. So we need maybe some citation tracking after retracting their retraction of papers and do a more complete and responsible work. Also, there are new functions that we can add to the system. So right now we are not doing single image analysis in terms of detecting splicing or when you are like removing pieces of an image. So we are doing only copy move, which is replicating content of an image within itself. But you may have cases like this where somebody just removed some content, and yeah, there are some tools that look into the noise of the images and they will figure out, okay, the distribution of the noise here doesn't follow the rest of the image. So definitely there's a mess that I can generate and point out that this region was modified. So yeah, there is from forensics tons of different solutions for doing these, and we need to add this to the system. So, and when I say add to the system, add a module and also provide annotations. And yeah, there's a lot. I can point to you many retraction notices that we can use for doing this. Other things that we do, so we are now in the times of synthetic content generation, and we had just last year a paper that was led by Politecnico di Milano by Dr. Mandelli, where we showed that it's possible to generate synthetic Western blots that are really hard to see that they are synthetic, and you can even like say and use a template and define where this thing should appear. And I don't think folks out there are doing it right now, but it will happen soon. So we need detection capabilities. And we had this first paper where we have one class classifier that can detect when a Western blot is pristine and not generated by an again process. So yeah, we need more research in this regard, and we can extrapolate this to microscopy and other types of images. Another thing, we are in times of chat GPT, so some text might be generated and we need to figure this out and also detect generated text. So far we worked only with images, and there is all the space of text that can be worked with. And also space for chart processing. So I just saw last week in Netflix, that is the show now, The King of Clones. I don't know if I had the chance, but it was, so this is about a South Korean researcher where he had two papers that were retracted because there were situations of results that were cloned. And the show, by the way, it misrepresents, given an idea that it was on the microscopy that the images were manipulated. But then when I provided these papers to our system, I could not find any problems with the microscopy. So after looking at the retraction notes, I just figured out that actually the problem was not with microscopy, but with the graphs, with the charts that the paper had. And if you look here, actually, these two lines, they should have spikes in the same position, but actually they should not have the same shape. Because here in this case, this is a paper about stem cell research. And what they are claiming is that they cloned the cell, and these spikes are like, say, the representation, manifestation of DNA or something like that. And the spikes should be in the same place. But what is happening here is actually that we have the same graph. And it shouldn't be the same graph. Only the spikes should be in the same place, so it shouldn't have the same shape. So this is why the paper was retracted. And yeah, so far our system is not able to process that. So we need some solutions for doing that. And finally, paper mill detection. As I mentioned, we are now seeing hundreds and maybe thousands of papers being published automatically, being fabricated and submitted to all sorts of conferences, and some groups are even selling authorship. And as you can see here, we started looking into this problem, and from 2010 to 2021, when you look at reasons for retraction, you can see that paper mill, which is the one in blue, has increased a lot lately, together with image duplication. So we need to deal with paper mill detection. And right now we are doing some research with Unicamp back in Brazil for improving provenance analysis to give support to paper mill detection. Okay, so yeah, this is all the future work we have, and that's amazing space, and we need collaborators. So we're looking for collaborators. We hope you have some interest in doing this. And that's yeah what I had for my talk. So I'm happy now to take questions and discuss all the things that I just presented to you.